As you know, there was, I'm sure all of you are familiar with this ST398, right, or clonal complex 398, that I'm not sure if it's called it emerged, but at least recognized in the Netherlands in 2004, around that area, 2005, IPVS, we've been discussing in Denmark and different places, so that since then, this MRSA has been named as livestock associated and reported from different parts of the world. So we did some study in the so I'll give you some examples using the tools that we use. So sequence type, or you see CC here, is for the clonal complex. What's the difference between sequence type and clonal complex? A little quiz based on what we discussed. When you have an isolate done by MLST, you have sequence type for each isolate, right? But when you group them as a population, you will get one or more clonal complex. If they are all related within one, you get one complex. Or you may get different sequence types that are highly distant, you may get two clonal complexes. But there is now one clonal complex known to be associated with livestock, particularly pigs, based on the initial report Netherlands and then different parts of particularly Europe. This is based on multi-locus sequence typing. It is ST398. It's unique. Initially it was considered non-typable by standard PFGE, but now many labs, including ours, we can do PFGE typing. Because why they were not able to do it initially was they used the classical restriction enzyme SMA1, SMA1, but for this particular strain, SMA1 does not cut it. It's the isoschizomas, the same sequence, but a different way of cutting. XMA1, the one I just highlighted here, can't cut this strain. So you can do positive gene electrophoresis as well. And then there are others, SPA typing, for example, like many groups do. So there are different ways of genotyping of MRSA and this particular strain. In the Netherlands, it's believed to be and reported to be circulating in 20% of the Dutch population. It's very high and particularly associated with pig farmers, families, I may have some data, and pig farm workers. I think I have it on maybe in the next slide. So one of the questions is the food products, meat or retail meat, can it carry MRSAs, has been always a question. A study from, I think this was a German study, DIBOR 2009, showed close to 12% carriage of MRSA from beef, veal, pork, chicken, others. You see this 11.9%. So it's fairly common. This is in Germany, not necessarily in the US. In the Dutch, in the Netherlands, community health study reported by Cooney in 2009, the same year, pig farmers, they're very high percent, family members high, veterinarians, veterinary family members close to 9%. And then in the study it says that they used also negative control, school children, but there were 3 positive children and they were all lived on pig farms. So really not so good negative control, but still it shows the extent of association pig farms. But that's again a European case, not really throughout the world. One of the questions was also, is the ST398 associated with antimicrobial growth promotant use? And clearly, this is a nice graph, in fact I borrowed from Peter Davis a few years ago when we were discussing about this issue, is, as you know, that Europeans started with Denmark, voluntary recall, and European Union, there is no antimicrobial growth promotant use, right, at least to the extent you can interpret it or interpret it, and to my knowledge there is no antimicrobial growth promotant use. since 2000. We started finishers later on in nursery, but this was detected in 2004, even after that. I don't think there is any relationship between the two, but if anyone wants to relate it, it's it's a reverse. Antibiotics were withdrawn, this ST398 emerged. It must be lots of other things to do, but again, this is not the main goal for this particular presentation. However, still there is a lack of good data in the United States, but there have been few groups, including ours, did a study looking at the molecular epidemiology of MRSA in porcine origin, generally MRSA. If there is ST398 or others. So quickly show you a few slides. So in Ohio, we used 10 courses of study. In fact, we did 3 studies. One is with Minnesota, Iowa, and North Carolina. That has been published recently on PLOS 1. This one was a little bit different study. The other one was looking at farm workers and pigs, mainly basing on nursery farms. This one is to look at from farm to retail. Is there MRSA in pigs? If so, does it persist from the farm to slaughterhouse, maybe to the retail pork? That was our main question. Okay. So if we have to do this, if we just use Staph, Aureus, and Methicillin resistance testing, that will not be conclusive even if you get MRSA throughout, because MRSA you get on the farm could be totally different from MRSA you get on the retail meat that may come from slaughterhouse worker and others. So we picked 10 farms, you see here from 1 to 10, and we followed them from farm to lairage in the pen where they stay at the slaughterhouse, to carcass swab, and retail pork. Retail pork is usually practically very difficult, right, to follow a pig to its meat in the retail. But really in Ohio, I really appreciated the packers. They were very collaborative, and the packer we're working with, they said that absolutely we can do it. Which ones do you want? Tag them, get them to slaughter, to carcass, and they tag them all the related sirloin, and then they ship that particular sirloin from our pigs to a known retail store with the boxes labeled for our study. The next day we go and buy from the retail. Usually it's practically different, but it's been very nice for us, it worked. But quickly the results. So if you look at cohort number 3, you see some MRSA out of 24 pigs, 3 positive. At lairage, as a slaughterhouse, you get higher positivity. I don't know if it's mechanically, remember it's nasal and oropharyngeal swabs. 11 of them gets positive, about 45%. Then on carcass it gets back to low. We found in retail 2 of them positive, 2 of the sirloin. But in all other cases there have been some positives, some negatives, a combination. Okay? When it comes to persistence, can you say that the strain persisted like on farms? If I ask you, if you look at these 3 pigs and 2 retail product has MRSA, can I make a point that the strain persisted from farm to retail? Yes or no? I guess I'm shaking heads. You agree? Yeah, I agree. I cannot say based on this data, based on this information. What is missing? What are we missing? If I have positive here, positive, why not? I say, oh yeah, still from the farm to retail, look at MRSA, it's persisting. Yes?
Is that the same pigs all the way across?
Yes, they are the same 24 tagged, and the meat is the same from the same pigs.
Same positives?
Yeah, I cannot say number 1 is the same pig within the 24 positive letter, but it's a group. Okay, number 2, whether the strain characteristic phenotypically as well as genotypically, I don't know at this stage. Okay. But quickly, we're dealing with molecular. Let me focus on the molecular. I had antimicrobial resistance data. We now have enterotoxin, which almost all of them don't have enterotoxin gene, just simply in a PCR. But we did multi-locus sequence typing. In fact, a lot more numbers now, but when I compare this, the results are still the same, more isolates, but we found 5 different sequence types. The most predominant we still consistently see is sequence type 5. Okay, overall prevalence was low, but it's significantly important in pigs and products. But sequence type 5 seems to be the predominance that we see definitely in Ohio. I cannot really speak for United States. And we found it in 2 farms. And we found it from the nasal, perianal swab, and one from carcass. The other one was sequence type 398, the livestock associated we discussed. It's the second one, fairly common, the second one, and we found it from 3 farms. Again, it was these 2. Farm number 3 had more number of isolates, but what we found was from nasal on the farm, perianal, and retail meats. There was 1 on retail meat as well. And then others, sequence type 9, 39, 72, they've been reported from China, from different places. Again, no causal relationship, but we know that it's genotypically similar ones. This remains the trend. I think even in the publication it's a lot more isolates, but sequence type 5 is that I didn't show you, but this is not a good graph, but I could have shown you more better on the phylogenetic, but it was more clustered. Okay, they were more clustered, but this is an example of sequence type 5, the predominant one, from layrage, holding pen, from pre-chill carcass, and from retail meat. And essentially identical on pulsed visual electrophoresis. This is more the indistinguishable nature. But also you see some variation. See, the antimicrobial resistance, I would not, this is the antibiotic resistance here. You cannot use it for subtyping here. Why? We purposely selected oxacillin resistant ones. So the goal is just to study methicillin resistance, so you need to be careful. If we just did, in this report, any Staph aureus, and then test for antibiotic resistance, you could use it for phenotyping, subtyping purpose. You see the difference? You can isolate Staph aureus any, and then you could do a methicillin resistance and other results to use it for study the resistance as well as for subtyping. But these are purposely, the media we grew them were purposely selective with Orsaga, oxacillin resistant Staph aureus agar. So you cannot make a point. But the point is, there has been some diversity with the PFGE, but you still, similar points we were able to make similar points with the MLST. Okay. This is, you can zoom into the genome. The point I want to make here is there is general genotyping to see strain relatedness, and then you can zoom into specific areas. In this case, this is a chromosomal cassette area where methicillin resistance is encoded. You can look at what types of staphylococcal chromosomal cassette types, SCC types. And many of the swine ones were not typable based on that. They were unique, new types. There are up to about 12 types right now, but they did not conform with any of them. The ST398 and others. So it seems to be unique, but we had some other information on that. Okay. So this is kind of a quick snapshot of